name is Amber. I have both an MD, so a Doctor of Medicine, and a Master's of Public Health. That was a part of my training. And I chose to do this, this science package with you guys because I actually initially didn't think that I was going to go to medical school. My initial plan was to become a brain researcher. I went through college. I got a volleyball scholarship, so I went to school out on the East Coast, and while I was there, I really got jazzed on this specific topic, brain reward and addiction pathology. I worked on a lab for a period of time after college, and it was at that time when I was actually doing brain research that I realized that I was probably a little bit more interested in the medical side of it than in the research side of it. I loved the research realm, but I also really liked working with people. And I was coaching volleyball at the time still too, so I thought that that might actually make for a good process of going into medicine, because I would get to do both of those things. I would get to do both science and I would get to do some coaching. So that was a little bit of an atypical path to medicine. It meant that I didn't go into medical school until I was in my late 20s. So I was 28 when I started. I then did a Master's of Public Health while I was there. And then I started doing my pediatric residency here at Seattle Children's. And I think you've had a couple other people who've presented from Seattle Children's. And then I now work as a pediatrician. So while I don't do this specific type of research anymore, I think that having a foundational knowledge about it is so helpful, because I may not work with, I work with some teenagers who are addicted to substances, but I also work with babies who are born to moms who are addicted to substances. And I think having some of this background information is really helpful for me as I practice medicine. So with that, let me just take a quick pause and make sure, does anybody have any outstanding questions right now before we just go ahead and get set up with the experiment? Are we good to go ahead and go? Okay, great. I'm getting a nod from Paul. Thank you, Paul. Yep. Okay, thanks, Ronnie. So the first thing in looking through this handout, which is really cool, I didn't do anything like this in high school. This is pretty awesome. Is to flip to page 5. I know it's a reverse image when you guys look at me here, but basically let's go to page 5, because we're going to set up our experiment. And we're going to do the same thing that you all have, which is take this little test strip, is what they call it. And I'm going to add, per the directions. So it says, obtain your 4 test cup strip from your kit. We're going to leave the first one empty. We're only going to use the 3 beyond it. And we're going to add 1 drop of dopamine indicator. So that's the blue looking material, to each of the small cups on the dopamine test cup strip. And you're going to use the pipette particularly that's labeled dopamine indicator. So let's all do that together and get set up. And I should probably say that you don't want to touch this stuff. You don't want to put it in your mouth. You don't want to get it on your clothes. So make sure you're working on a stable surface. I'm going to recap it. And then I've got like a little box over here. I'm just going to put all my dirty stuff in. Okay. So does everybody have 1 drop of your indicators inside of each of your 3 little test tubes there?
We have a question of just 1 drop.
Just 1 drop. Yep. Thank you. Just 1 drop of the indicator. I'm just going to double check this. Yep, 1 drop of dopamine indicator to each of the small cups.
And which indicators is another question?
It's called the dopamine indicator, and it is a blue colored, it's the only colored material that you have. Bluish greenish color. Okay. So if everybody has that.
To each cup or just 1 cup?
To each, we're going to do them to 3. So we're going to, you can kind of see my first one here doesn't have anything in it, but then my next 3 each have 1 drop. Okay. And then once we've got that set up, we're then going to add our, the bottles, whoops, sorry, labeled 0 minute. You should have a test tube labeled 30 minute. And then a test tube labeled 60 minute. And in addition to that, you have then these little tubes that are called pipettes. And each pipette also has a label on it that says 0 minute, 30 minute, 60 minute. So we're going to use the associated pipette with the associated test tube. So I have here a 0 minute test tube filled with fluid and a 0 minute pipette. And you're going to take 6 drops of the 0 minute and put it into the first little test tube here that has your color indicator in it. Okay. And you do want to make sure in any experiment that you're not cross-reacting things. So that is why you want to use specifically the one pipette with the one test tube. Once we're done, we're going to put them off to the side. Then we're going to take our next ones, and we're not going to reuse items. Okay, so I've drawn up some liquid.
Looks like there's a little bit of confusion.
Okay.
If y'all don't mind clarifying what's the confusion,
Yeah.
like that, and we can work through that.
Is it easier if I hold it up and then people can kind of see at the same time? Yeah, go ahead, Ronnie. Can you unmute yourself?
Okay. So I was wondering if we're mixing with the dopamine or putting in a separate cup.
Sorry, I missed a little bit. Would you mind, sorry, just speaking up just a little bit?
Are we mixing with the dopamine or are we just putting in a separate cup for the 0 minute?
We're going to mix it. Yeah. So each one of the indicators, the indicator drops, the green drops that you put in there are going to stay. Can you kind of like see right here? Right. And then I'm going to take my 0 minute test tube material and I'm going to put 6 drops. 1, 2, 3, 4, 5, 6. Okay. And you can kind of see that there's a color that came out of it, right? If I were to hold this up to your color chart, which I think you guys have, that 0 minute corresponds to the color yellow, would you say? So we can make a note that this one is the 0 minute. Okay, but I think what we'll get a better sense of is when we then take our 30 minute, we're into all of the 3. Thank you, Sarah. No, you're just going to do it to the one test tube. Okay, so I will then take the 30 minute, draw up my material, and now watch. I put it not into the yellow one, I put it into the next adjacent one, and I put 6 drops. 1, 2, 3, 4, 5, 6. And that's off to the side. And then I take my last one, which is the 60 minute one. I'm just going to double check 60 minutes on my pipette, 60 minutes on my liquid, and I pipe this in. 2, 3, 4, 5, 6. Great. So that's what everybody should kind of start off with. It's going to be helpful once we kind of go through this process, and you'll kind of see why the different colors are there. But once we have that, then we can kind of set this off to the side. Then 30-minute into the one adjacent, correct? Yeah, so the 30-minute goes into this middle one. Then 6 drops of your 60-minute are going to go into this last one. Okay, and so then we'll leave it off to the side, and then we'll come back and we'll kind of look at what those colors mean and why we get a different color as we've gone through that. The colors of the mixture look pretty, I agree. Okay, so why don't we just pause right there for a few minutes? We can answer any other questions, just to kind of make sure that we get all that set up, and then we can talk about why we're seeing different colors show up, because I think that that's going to be really informative as we then talk about dopamine and drug addiction and how the brain changes just a little bit when you have exposure to those substances. Okay. Great. Can I just get a little show, maybe like in the chat? Is everybody have that all set up and are good to go? Mm-hmm. Great. Okay, good to go. Great. Okay, so moving on. So now let's go back to the very first page then and talk about our patient. Well, I would call him a patient. We'll call him, this is just a medical report. So are you okay if I go ahead and read this out loud? You guys just go ahead and follow along with me. So Ray is feeling horrible. He is very depressed and nervous. He has a headache and nausea, and he's having trouble sleeping. He's experienced some of these symptoms in the past, but they disappear when he uses something called Floritrip, a mixture of over-the-counter and herbal medications. Ray doesn't think that his symptoms are caused by Floritrip addiction. He says lots of people use Floritrip. It's not illegal. It's not addictive. What's wrong with feeling good? He likes using Floritrip because it makes him feel happy and confident. And he says, quote, I only take it when I'm feeling low or stressed out, a little Floritrip, and I feel great. He's been using Floritrip for about a year, but over the course of the past few months, Ray's use of Floritrip has increased from once a day to more than 5 times a day. He's not been able to use Floritrip though for the past few days because he ran out of money, so he can't buy it anymore. And he was recently fired from his part-time job because he skipped work a lot, especially after late nights of doing Floritrip with friends. So that kind of sets our scenario. So let's say, for example, Ray is 17 and he's sitting in front of me and we kind of talk about this and we talk about the different things that have been going on with him using this Floritrip. So then this next question down below is based on what you've heard about from Ray so far. Do you think that Floritrip, even though he says it's not addictive, do you think that it's addictive, even though it's legal? So it's not technically illegal. And what might be helpful is that just above that, there's a quote that says that drug addiction is defined as uncontrollable compulsive drug seeking and use, even in the face of negative health consequences. Great. Okay, so we've got some folks in the chat. Yes, it is. Yes. So can some people write in why they think that? I'm telling you that. I'm kind of leading you there. But what, based on the definition of addiction, do you think that he's showing signs of? Great. He went from using it once a day to 5 times a day, right? So the technical term, they use that because it makes him feel good. He uses it often. His body stopped producing the neurotransmitter, and now he's relying on Floritrip to create the feelings. Okay, gotcha. I hear you. Right, so the first thing that I noted too was that his use has increased. So that's a definite sign that his body has become accommodated to some extent. He gets used to the one dose that he was at, and he needs higher and higher doses in order to feel good. He uses it more often. Okay. There's something else. Do you remember how he hasn't been taking it lately because he ran out of money? even though it is something he can live without. Okay, he ran out of money. Yeah. The withdrawal symptoms, exactly. So his body has now become used to having it around, and his body is accommodated. And so now that he no longer has it in his body, he starts to show withdrawal symptoms. So that's another sign of addiction. And then the last one that is kind of key with Ray is actually from, you can kind of get the sense from the quote. He was skipping work to use it, right? Are we supposed to use the goggles? I don't have goggles. Sorry, Sarah. Yeah, so one of the things when people kind of sit down with me, or when I talk with, say, folks like in the emergency room, if people are having to, or if they're suffering consequences, right? They're spending time in jail, or they've lost their job, or they're starting to steal because they need to do things in order to support that, that addiction or that craving that they have, then that's one of the ways that we think that this is interfering with their lives, right, in a significant capacity. So even in the face of negative health and social consequences, he can't help the fact that he's using more and more of the FloRitrip. Great, okay. So now flipping over onto page 2, we can kind of talk about the science behind it. And what we're talking about specifically is something called the reward pathway inside our brains. And one of the reasons that I was so curious about this in undergraduate school and college was because there's a lot of addiction in my family, a lot of alcohol addiction. And I couldn't quite understand why people couldn't change their behavior. Why couldn't people just go to work and do the things that they were supposed to do, right? And so this was kind of really mind-blowing for me to understand that it wasn't really a personality flaw, right? That somebody wasn't able to do those things. There is a wiring system inside our brain called the reward pathway that we're going to talk about that basically makes it so that really good things like alcohol or like sugar or like really awesome feeling things increase your level of dopamine, and it starts to reward that pathway in a way that makes you want to choose to do those things, even if they're not necessarily the best choices for you. So the specific details talk about an area of the brain called the nucleus accumbens, which is in what's called the primitive area of the brain. If you think about it, I can kind of show you your primitive area of the brain is kind of like if you make a fist like this. This is the core of your brain. Primitive. Thank you. What does a primitive mean? So primitive means an earlier version of our brains, the brains that we basically still kind of share with animals. One of the reasons that we can do addiction research using rats, which I don't know if anybody thought of how weird is that, right? That we study rats, is because we all share a common core brain with cats and dogs and chimps and rats, with all mammals. So your primitive brain is a structure that, if you think about it, is kind of like a small little fist. It just sends signals back and forth that talk about things that feel good, things that make you scared, and really just makes it so that you just live every day. What humans, primates have come to evolve is a cortex brain that sits around kind of like the outside. And this is sort of like, if you look at the pictures of a brain, which I know that you've all seen, the brain that sort of has like the curves to it, right? They go in and out, the sulci and the gyri that sit around the exterior portion. The exterior portion is your evolved brain, and the core part is your primitive brain. So the reward pathway that's built out of the nucleus accumbens, the ventral tegmental area, sits in the core part. And this is where those signals, those dopamine signals, get sent from the sending neuron to the receiving neuron. And dopamine then gets released. And that becomes what's called a reward circuit, such that if I eat or drink something that increases my dopamine, then the receiving neuron senses that, and it says, oh, I like that a lot. Give me more. I want to do more of it. And so if you take things like, we know certain things that are addictive, cocaine, alcohol, certain foods, and increased meth, great, okay, sure. and increase that signaling that starts to occur, you get more dopamine, you get a dopamine flood. And because the neurons that are on the receiving portion say, hey, that dopamine is really cool, I'm gonna want more of it. And it wants to draw more and create that circuit that's even more reinforced. And that then starts to drive our behavior, because we want to seek out more of that really good feeling, because we're animals and that's just kind of what we want to do. So with that, if you guys read sort of through this, information, it's the pleasure sensation is kind of the other way that we think about it. There are questions on page 3. So what is the function of the reward pathway of the brain? Why do we have it? Alexa said to help us regulate our dependence, to not feel depressed all the time, to have a pleasure sensation. Good. Okay, Sarah, you're on the right track. So we think that this process is there because if you're a little animal, let's think we're like little squirrels or like little rats in the land before time, and you're going along and you find something, and let's say that you find like, I don't know, a tree or like a little flower, and it tastes really good, and you eat it and you're like, wow, that's really, really good. And then you go off and you look for something again. What you eventually want to be able to do is you want to be able to remember where that little flower was, where that delicious little acorn was. And so you get this really good feeling of dopamine, and so that you kind of cue in and say, oh, I want that again. I need to remember how to get to this place so I can come back and keep getting this really good food. And we think that there's a similar association with sexual reproduction, right? There's supposed to be a rewarding feeling that goes along with it so that you keep doing it, so that you keep your lineage coming along, right? Keep making rat babies and little squirrel babies and little human babies, etc. So we're meant to have a pleasure sensation. If we didn't have it, you're right, we would be depressed all the time. Why would you get up and get out of bed, right? Why would you go to work? It wouldn't work. So we think that there's this process inside of us that's meant to help us further ourselves along as a species. And so that's why even the primitive brain, right, that central core brain that we share with animals and cats and dogs, they all have this same principle. It's this pleasure principle. And so that is meant to drive our success as a species. And so then the next question is, list 2 behaviors which cause impulses to travel through the reward pathway in the brain. And so we kind of talked about it, right? Eating and sexual reproduction are 2 well-known, well-documented, well-established ones. And we think that, again, that's just sort of, that's innate to us so that we keep doing those things, so that we keep evolving. Then question 3 is, what causes animals to repeat behaviors that stimulate the brain reward region? So why does an animal keep doing it? I think I kind of already talked about it, right? It's because you do have specific neurotransmitters. Have people heard of neurotransmitters before? I would assume so. Yes, great. There's more than one kind, right? So there's dopamine is a neurotransmitter, which we just talked about. That's going to be the focus here. There's another one called GABA. There's another one called serotonin. You probably have all heard of that too. And there's a handful of other ones that are all being discovered as time goes on. And so we think that there are some things that all just, it's called neuromodulation. It's the ability for our brain to rewire in the presence of some of these neurotransmitters. And so some of them make your system go on high alert, and some of them make your system get relaxed and kind of chill out, probably for the exact same reasons around fight or flight and being really scared. But we know that addictive drugs specifically activate the brain reward pathway and the neurotransmitter called dopamine. And then one of the other questions that I think is important here is, do you think that shopping, smoking, gambling may activate the reward pathway and lead to addiction for some people? What do you think? Yes. Okay. Why do you think that? This is a kind of, well, it was anyway when I was like doing this research like 15 years ago. This was kind of controversial for people to think that that something like shopping could be an addiction. Why do you think shopping's an addiction? Whoever said yes. Right. Okay. So some people might actually really feel like that happiness and pleasure that kind of goes along with it. Does that happen to anybody? Like you buy something and you're like, yeah, I'm really happy. It lasts for me for all of like 10 seconds. Yeah. Nods, some head nods. Yeah, sure. It happens. You're right. And one of the reasons we think that people get addicted to shopping, right, is that you kind of keep looking for that feeling. So you get that sort of burst. You're like, yeah, that's a really awesome thing that I just bought. And that feeling lasts for like a period of time. It kind of goes away. And then you feel like you need to shop more, right, in order to kind of get that. And then sometimes you do it to the point that it's actually destructive, right? People may run up their credit card bills. Sometimes people buy things that are not needs but wants. And that demonstrates to us that it probably is, it has an addiction component. Ronnie, right? Sometimes you do it and you regret it after, but then you do it again. So you lost a little bit of control, right? Everybody likes to think that we're completely in control of our behaviors. And what neuroscience has shown us is that we're not always in control of our behaviors. Like sometimes we're doing things because we just want to get those good feelings again, right? Super, guys. Okay, so the parts of the rat brain, so now I'm looking at question number 6. This is a good question. The parts of the rat brain involved in the reward pathway are shown in the diagram on the right. So we kind of talked about that. The nucleus accumbens, the ventral tegmental. You can kind of see that on this diagram, right? The primitive portion of the brain looks different than ours, but there are the exact same structures that are still there. Some scientists who study the effects of addictive drugs on the brains use rats instead of humans in their experiments. What are 2 advantages of using rats instead of humans when conducting research on the effects of drug on the brain? What do you guys think? Why not use humans? It may harm us, right? Yeah. Their brains aren't formed well as humans. Actually, ironically, the rat primitive brain is actually very similar to ours because it would be dehumanizing. Yeah. There are these ethical dilemmas, right, when it comes to like doing research. And so, yeah, Alexa hit on it, and Sophia hit on it, is that if we're testing something and we don't really know whether there's a good response or a bad response, then you can't necessarily use a human in that scenario. You know, we feel like we feel like we can only do that if we feel like there's a better risk benefit scenario, so more benefits than risks that are associated. But for the most part, you're giving a brain or giving a drug to somebody and then testing them for it. Well, honestly, those were studies that were done a long, long time ago. Humans were used in those scenarios. They're not anymore. Another thing to keep in mind is that you can do these studies faster by using a rat model than using a human model. And so especially for things when it comes to like laboratory sciences and spaces, people have to kind of think about the cost that's involved. And generally you can get the results a little bit faster. Okay, great. Okay, so let's now talk about part 3. So now we're going to kind of get to the part where we can kind of start to look at the specifics of the experiment that we set up, right? So what this scientist does, and these are actually science studies that I did when I was in college. I want to determine, if I'm the scientist here, I want to determine if fluorotrip is an addictive drug. We think that it is based on some of Ray's behavior, but we want to be able to prove it. And one of the ways that we can prove it is by demonstrating that the dopamine levels are higher in the brain after somebody has taken fluorotrip than if they're not. So what we'll do is we will take a look at 3 different rats. We're looking at the brain substances of 3 different rats. Rat A that was given sugar, We're calling that one basically the control. Rat B, which is given cocaine, and then rat C, which was given fluorotrip. And what we're doing, right, what we're saying is that hypothetically, these animals have access to these different drugs. And then what we're doing is after we're taking small little samples of their brain fluid at times after the animal has access to the drug. So rat A is just drinking some sugar water. At 0 minutes, so right as soon as he gets the sugar water, we take some samples. We take it again 30 minutes later. We take it again 60 minutes later. And so what you can see is that they've already done the results. They've already given us the numbers, the concentration of dopamine in the rat brain for rat A and rat B. What we're going to do is we're going to look at rat C, which one is given fluorotrip, because this will allow us to see whether fluorotrip is more similar to control, which is sugar, or more similar to known addictive substance, which is cocaine. Okay, so let me grab my, everybody have their substances. We can now look at the 0 minute mark, which was this little one right here, the first one next to my finger, which is yellow, right? And would everybody kind of agree that the yellow one is 10 on their chart? So you would put 10, yes, great, okay, in that box, yep. And then I liked what Sarah just said, the 30-minute mark is actually closer to, which number did you guys get on this one, if you compare it? The 30-minute mark looks more like 400, right? And the 60-minute mark looks more like 100, okay? Everybody got those same numbers? Great. So now it's just a matter of comparing fluorotrip to rat A given sugar, rat B given cocaine. So if our hypothesis was, we believe that fluorotrip is an addictive substance based on how Ray has been acting with and without fluorotrip, have we confirmed that? Yep, I think that yep is for me. Yep, cocaine and fluorotrip are the same, right? Very good. So fluorotrip is acting just like cocaine. We know that cocaine is a known addictive substance. So we've now proven, at least in that study, that fluorotrip acts like cocaine, which is an addictive substance. Great. So based on your answers, what happens to the level of dopamine in the brain when rat is given sugar? Is it 0? It's not, right? There's some dopamine released with virtually everything that we do. Sugar's good, right? I mean, who doesn't think that sugar's good? So it tastes good. It's a rewarding substance. You get a little bit of a dopamine surge when you eat sugar, when you eat candy, you know, similar to kind of like when I, you know, eating shopping or eating other foods and like going shopping, you get like that little bit of rush and then you kind of get that feeling that's, you don't feel so good on the backside. So then you want more of it. So sugar is a known addictive substance. You know, 15 years ago, 20 years ago, nobody would have said that. But we know that that's the case now. So yes, dopamine goes up. It goes up a little bit. But when you compare the levels of dopamine in the rat given fluorotrip with the levels of dopamine given sugar, we know that dopamine is much, much higher, right? On the matter of anywhere between 40 to 10-fold. Compare the levels of dopamine. Dopamine is the same as cocaine. And we think that fluorotrip is addictive. Great, we blew right through that. Okay. Let's pause for questions. Any other questions right now? Is any of this new or old news for you guys? Like, how do you feel about some of the stuff that I'm saying in terms of maybe you've heard it all before? New, great, okay. Alexa, ooh, deep, man. That's a deep question. Could potentially anything become an addiction? What do you think? Yeah, Ronnie says so. Okay, a little background from child psych class, or from psych class. Great, okay, good. Okay. Well, let's talk a little bit more about this second experiment, because this one's pretty cool. I've done some of these experiments before in rats. And what this one kind of shows, so the first experiment just literally shows that there's more dopamine in the brain when somebody takes a substance, right? But the next phase of proving that that substance alters your behavior is to show that rats, like humans, act different when they have different availability of substances. So that is what this next study looks like, this next experiment. So page 7, does everybody kind of understand the setup behind this experiment? We're talking again about the 3 different rats. Rat A, who's having exposure to sugar. Rat B, who's having exposure to cocaine. And rat 3, who's having exposure to Flortrip. So you can kind of picture these rats are hooked up in their little individual cages. They have right here, they've got kind of an IV going in. So they've got kind of like a little cannula that goes into their brain. And in that room, in that little cage that they're in, there's something called a lever that they can push. And how many times the rat pushes the lever gives you an indication of how much their brain is liking or is being stimulated by what it is that they're receiving. So the more reinforcing something becomes, right? As soon as the rats start to realize, oh, if I push this lever, I get more of this really good stuff. Well, then the next times that you put the rats in those cages, they push the lever more times and they do it faster because they've learned. They've had a behavioral association and they've learned if I push this, I get that stuff that makes me feel really good. Okay, Pavlov. A little bit. Pavlov did, so there's 2 kinds of conditioning. There's operant conditioning and there's stimulant conditioning. And this one's a little bit different than Pavlov because we're giving them an action to perform, right? Whereas Pavlov's dog salivated merely in the presence of something else. You kind of didn't make him change his behavior, but yes, same lines. This is all called conditioning. So flipping over, page 8 is a data table. And so what you can see, and I've kind of already filled this out for you. I don't know if you guys did this too at home. But if you make a plot of time on the x-axis down here, the lower axis that runs across, I put that time in days. And then I put the number of lever presses on the y-axis. Then what you can see is at day 1, rat A, who's only getting hooked up to sugar, he pushes the lever 4 times. They put him back in the next day, he pushes the lever 3 times. A couple days later, he pushes it 4 times. So that's what all of these things are down here. So I can plot over time how reinforcing sugar is on this rat. What did you make the scale? In days, I use this scale just as the days. Was that the question, Sarah? On the number of lever presses, I looked to see how many lever presses we had. So we had a high of 77. So I used each little tick is a 5-point increment on lever presses. Can you guys see that a little bit? Should I pause or should I keep going? Okay, great. So then if we look at rat B, you put rat B in the box on day 1, he pushes the button 3 times. On day 2, he pushes it 7 times. Day 3, he pushes it 12 times. By day 10, he's pushing it 29 times, then 52, then 77. So he basically, right, he gets put in that box and he's like, I know exactly what I'm gonna do. I'm gonna push this lever over and over again so I can get as much of that stimulating material into my brain as possible. Okay, so what I haven't done on this last one is plot the flora trip. So if I were to do that, day 1 of flora trip produces 4. So I put like a little mark there. I've got a blue marker now. Day 2 is 8. Day 5 in here is about 10. So this probably isn't news to anybody, right? But this is going to end up looking what? Like cocaine. So day 10 goes up to 27. Day 15 goes up to 49. Day 20 goes up to 73. So if I make my connections, flora trip looks just like cocaine from a behavioral perspective. So we've now got two things, right, that are correlating flora trip with an addictive substance. Not only does it raise the dopamine levels in your brain, just like cocaine does, it also makes you act different in the presence of it. So then if we go to page 9, the question that shows up is, what is the independent variable in this experiment? With the definition being the variable factor that you change, and it's usually associated with the if part of the hypothesis. This part's kind of tricky. The dependent variable or the responding variable is the variable that may change as a result of the independent variable. So did anybody have any thoughts on independent variable? Would the independent variable be the amount of times the rat presses the lever? What do you think? I was thinking that that one might be the dependent variable. That's the response, right? So the independent variable is, to me, what the rats are exposed to. But the dependent variable is then the changes, the responses that then get made in order to increase their exposure. So rat A and rat B were controls in this experiment. They provided a basis of comparison. Okay, good. So which rat provided a control? Oh, I think I already gave this away one. To show the difference between a rat's reaction to normal rewarding stimulus and flora trip. So which one was the control? Sugar, yeah. And then which rat provided a control, a basis of comparison to show the difference between a rat's reaction of an addictive substance and flora trip? So I guess it's more like a comparison. Yeah, the basis of comparison would be the cocaine. Yeah. So why do you think the number of lever presses for sugar increase? They could have decreased. So that's just because it made them happy. It became addictive. Yeah, it probably became a little bit addictive. It was good. It felt good. It tasted good. We also kind of know that anytime you have a control, you're going to have some amount of margin or error. You're going to have some amount of, some rats may not push it at all for the sugar. And so you end up with kind of a little bit of variation that goes around. Standard deviation sometimes, right, is also what we'll call it when we do like our statistical analysis. But then when you then start to make your comparisons, you realize that there's a really clear distinguish between sugar at the bottom, right, and cocaine that goes up. So the one that would have been really tricky, right, is what if it would have fallen somewhere in between sugar and cocaine, right? So what if on the graph, instead of clearly aligning with cocaine, so it's kind of like a little bit of a giveaway. You know, if it would have fallen somewhere right in the middle, in between those two, that might have been a little bit harder to distinguish. So in that sense, you know, people just might have needed to do more studies, right? To make sure that they were okay with that clarification. Okay, so I think we're kind of getting to this point where we say, does the data from the rat lever pressing support the hypothesis that fluorotrip is addictive? And it does, yeah. So here's a question. I like question number 9. State one way that the scientist's experiment could be improved to make it more reliable. So now, you know, we think that we have these things. It looks like it's kind of a slam dunk, but what could scientists basically do in order to make sure that before they say with absolute certainty that fluorotrip is addictive, what other steps that could be taken? More test subjects, okay, right. You're right, Alexa, because right now we've got one rat who got cocaine and one rat who got fluorotrip and one rat that got sugar. So yeah, you could scale this up. So you could say, let's do 10 rats in each category, more species, okay? So you're right. So then sometimes what people do, more species becomes a little bit more controversial. So sometimes people will cross over and start to do cat and dog studies, which I think makes us all feel a little bit squeamish. Sometimes people will then also do primate studies. So yeah, cross it over and say, maybe, you know, maybe it's not just the rat brain that does this. Let's really make sure that there's some consistency across all of those, okay? Any other ideas? Those are good ones. Great, Ronnie, right. So what if you take the same rats and you mix them up so that you kind of, you know, you let them completely, presuming that there's no long-term brain changes, right? We don't necessarily think that there is, but there might be, who knows? You would then get all the rats back to neutral. You would then reassign them to the different categories. And then if the substance is really the thing that's making them different, then it should produce the same results again. Redo the experiment, right? So yeah, then that's something else that somebody else would do. So there's a lot of time in science where somebody does a study and the results look suggestive of a certain outcome. But before we go marching down, you know, a drug test or a behavioral intervention or anything like that, we get many other people from many other departments, many other facilities doing those studies, just frankly repeating them. So they set them up the exact same way and they just repeat them. And then they compare and they say, well, yeah, I got the same results or I didn't get these same results. And that helps to weed out certain kinds of biases, right? Because there's bias that goes into everything that we do. And we know that if I'm the researcher, I want to have an outcome. I want to make the assumption, especially if my hypothesis is, I think that fluorotrip is addictive. You know, we kind of all like to confirm our biases. And so one of the ways that you control for all of these different things is you repeat it, you scale it up, you resort the groups, and then you do another control trial again. Great, good, awesome. You guys are cool. How are we doing on time, Paul and Max?
We have 10 minutes left, or 11 minutes left. So there's going to be a little survey in the to give to you guys.
Okay, great. What other questions do people have? We've got, I mean, I think we hit the high points about, oh, wait a minute, sorry. Question number 10. Okay, this is really good. So let's say that, like me, you're a passionate physician, you're a pediatrician, you're taking care of people, and let's say that you get a lot of people like Ray coming into your office, right? Maybe over the course of like a year, you've talked to 10, 15, 20 people. And you're starting to realize that if fluorotrip really is addictive, if it's really messing with Ray's life. It's making it that he's losing his job. Maybe he's going to lose his home. Maybe if he has kids, he's not going to be able to, take care of his kids anymore. You now feel like you have to advocate, right? You have to go to the lawmakers and say, we need this to be regulated, right? And sanctioned in some way. So who can share with me, like, what would you say in order to persuade the lawmakers that something needs to change? You'd show the evidence, yeah. Show the data. Oh man. I've done this before. Yeah, I have. I'm a data person. I really am. Because to me, it's sort of like, look at this graph, right? It's so obvious. Yeah. Show how it's affecting them. Yeah, exactly. Exactly. I think that those are two really good things. You have to know the science behind it. You have to be able to explain to people and, kind of have an understanding of the scientific support, because science is real, science is evidence. We need all those different things. Lawmakers and legislators also really appreciate how it's affecting them stories, right? They really appreciate hearing from people that, that drug that's just sort of like sitting out there that you can get, at the corner store. Oh, it really is affecting people. I had no idea. And you tell them some of the stories and everything. But yeah, you have to have a good understanding of both in order to help legislators understand these things. So there was something called bath salts that people were using a lot of 5-8 years ago. I don't know if those are still going around. Kratom is another thing. You can kind of see it for sale, like in some of the corner stores and stuff. So these are actually a lot of very similar things that we're trying to, from a legal perspective, especially in the state of Washington, get people to understand that they affect people and they're not benign. It affects their brains. Then you could explain how it could affect them financially. Ooh, good, Alexa. I like where you're going with that. Yeah, I think you can let the legislators know how it affects them financially, right? That's what a lot of people really get drawn on, is the bottom line. And then Ronnie says, yeah, when it's making the government money, they're hesitant to listen. It's true. It's true. But sometimes, what we can talk about is if people get addicted to a substance and they lose their jobs and they go on unemployment, right? They need help from the government. And so sometimes what we like to convince people of, or try to convince people of, is even if you're not necessarily gaining any money in the immediacy, then preventing some of those outcomes is better for society as a whole, and it's better for taxpayers, and it actually costs less money overall. But you've hit on a big key thing, which is the difference between preventative medicine, trying to stop things before it happens, versus trying to fix something in the moment. It's a big deal. Cool. Well, so what other questions do you guys have? Do you want to hear anything else about what I do, doctor life, medical school? How hard is it? Residency? Do you guys know what residency is? That's like the training after medical school. It's fun. It's hard. But it's good. I like my job. How was your experience in medical school? My experience was great. I went to I'm from Arizona. And so after I was out on the East Coast for a period of time, I came back and I went to my home institution for medical school. And my school had a big emphasis on community health. So I did a lot of things where I was meeting and talking with people like almost immediately. So it was a small class. So for a medical school, it was only about 100. And so I got to know a lot of people really, really well. And there was actually a fair amount of us who did things like primary care, pediatrics, and family medicine. And Nico asked, did I have any scholarships other than the volleyball one? There were a couple grants that I was able to get while I was already at an undergrad, when I was already in college. But the volleyball scholarship was a big deal. That paid for all of my undergraduate education. So that paid for 4 years. And then I had a few other little grants that I did while I was an athlete. Ooh, they're all coming in now. What careers would you recommend for those interested in neurology? Great, yeah. Pediatric neurology, of course. So Yeah, neurology is great. There's also something called physiology, which kind of connects the body and neurology. That's sort of helping people like after they've had car accidents and need rehab and have like chronic pain and things like that. But neurology is pretty great. There's also neurosurgery. So there's a difference between neurology and neurosurgery. Surgery is obviously the part where you're operating on the brain. Neurologists take care of folks more with things like epilepsy and seizure. 5 minutes left. How did I get the scholarships? I was lucky in that I played for a really good club volleyball team that brought scouts to our tournaments. I didn't know a whole lot when I was where you all are. I didn't know a lot about scholarships. You know, my family, you know, didn't have a lot of money, single mom, and so we didn't know about anything. And so luckily somebody was looking out for me, and they brought scouts to me so that I could audition in front of them, and then I was able to get a volleyball scholarship. Is there a career that has a mix between developmental psychology and neurology? Ooh, you've got so much knowledge, Alexa. In the pediatric realm, there is something called developmental and behavioral pediatrics. I would say that that's probably one of the closest links. That's probably the closest, because that's going to incorporate a lot of neurology in it as well, because that ends up working with a lot of kids who have things like congenital disorders, cerebral palsy, and various other things that kind of help, you know, the cognitive and the neurological aspects of it. How about psychiatry? Psychiatry is great. Yeah. Psychiatry would probably not get you as much of the neurology if there was an interest there, but psychiatry is really, really focused on a lot of this neuroscience right now. And so I think we're getting a really good, a really much better understanding of the mysteries of the brain. Does pediatric neurology deal with things with data? Do pediatric neurologists deal with things with data? They do. So some pediatric neurologists still do research. They will still have a lab, and they might do some of the exact same rat-based research that you and I just kind of walked through right now. Other things about pediatric neurology will focus on things like epilepsy, and they will use medications to try to decrease epilepsy in some people. And sometimes those are clinical trials that you can only do in humans. And so that will have some data that goes into it. How long does it take someone to become a psychiatrist? Okay, so how many years of college? So you would have to do 4 years of college, 4 years of medical school, and then after medical school, you would do a psychiatry residency on top of that. And those are generally 3 years on top of that. So it would be 4 plus 4, which is 8 plus 3. So 11 years before you would then become a psychiatrist. There are a handful of combined programs. So for those of you who are interested in medicine, there are a handful of what's called fast track programs, where you do a combination of undergrad and medical school together. So that means that you still come out faster on the backside of that. But the additional training that you do after medical school, called the residency program, is anywhere between 3 to 8 years, depending on what you choose. And those don't have a whole lot of flexibility. Those are set time periods as of right now. So my pediatric training was 3 years. Things like a combined internal medicine and peds is 4 years. Things like general surgery can be 5 years. 2 minutes left. Do they do trial cases on Alzheimer's? Sure, yeah, happening as we speak. How long would it take to become a pediatric neurologist? So it would be, this one's longer. So it's 4 years of college, 4 years of medical school, 3 years of, well, it could be anywhere between an extra 4 or an extra 6 years. There are a handful of combined pediatric neurology programs that take 4 years. Sometimes what people end up doing is they do pediatrics and then they do neurology, and that's an extra 6 years. So it would be anywhere between 14 to 16 years.